Hello everyone. Telomerase enzyme structure, function and mechanism is what we are going to talk today. We first need to know what is known as end replication problem. During DNA replication of eukaryotes, we know we have linear chromosomes which have ends. So when the chromosome is replicating, the lagging and leading strands are being made. In the lagging strand, primers are recruited by primase, and then the polymerase enzyme synthesizes adding DNTPs to the 3' OH of the primers. Then the primers get replaced. So primers are removed by RNase H enzyme, and then they are being replaced by DNA polymerase as well. But this doesn't happen when it comes to the 3' end of the template of the lagging strand. Why? Because at the end, imagine this primer getting removed, but DNA polymerase cannot recruit DNTPs here as it doesn't have 3' OH to continue synthesizing the DNA that is corresponding to the part here. So what happens is the gap isn't filled and it therefore remains as a 3' overhang. We call this as a 3' overhang. So when the gap is not filled, the DNA damage system in the cell recognizes it as a DNA damage and comes and cleaves it. So nucleases are that does that. So if this happens during each replication cycle, the DNA length becomes shorter and shorter. So this would result in the loss of essential genes, which might be important for the survival of the cell. To avoid this, the telomeres, we know the end of the chromosomes are called as telomeres. Telomeres are non-coding DNA. They have their own protective mechanism. The special group of proteins that protects or helps on this function is shelterin. So it's a group of 6 proteins. So you can see TRF1, TRF2, TIN2, TPP1, RAP1 and POT1. So what happens is once 3' overhang is like this, the sheltering protein resides on it, and then this causes it to loop and hide the 3' overhang. So what happens is it forms a T loop. So also there is another loop called D loop. So this is done in order to not be recognizable by the DNA damage system and avoid being cleaved by the nuclease enzymes. The function of telomerase is also very related to shelterin because for the telomerase binding, shelterin is actually needed. Telomerase comes and binds the TPP1 protein which is in the shelterin. So we'll take a look at what telomerase looks like. So telomerase enzyme has two parts, which one is called as the TERT part. This is the catalytic protein. So we call it telomerase reverse transcriptase. So this is the telomerase RNA. So it has inbuilt RNA template. So if I draw the telomerase enzyme like this, it has two parts. So one, it has a template within it. This template contains RNA, a sequence of RNA nucleotides. So the telomerase enzyme, we know it is a reverse transcriptase. What it does is it has a RNA copy here. It would make a template of DNAs. So which where the telomerase would bind to this end and elongate it, making the DNA elongating the DNA strand. So these are the two paths, TR and TERT. So actually there are 4 domains of the catalytic protein as TEN, TRBD, RT and CTE. This is how the catalytic protein looks like. You can see there's a ring-like structure and the hole in the middle. They actually say that it corresponds to the right-hand form of us. So you can see the thumb, which corresponds to the CTE domain, the palm and the fingers, which corresponds to the RT domain subdomains, and the TRBD part here. Now we'll see about how telomerase is being catalytically active. Actually, telomerase is not expressed in all cells in the same amount. It's mostly expressed in stem cells and in the germ cells. So because they need to continuously replicate. But in the other somatic cells, telomerase are not expressed as in stem cells and germ cells. So let's see how telomerase is being recruited. The HTRT part is made in the cytoplasm and the HTR part is made inside the nucleus. So they both need to be assembled in order to make the catalytically active telomerase. HTRT is being recruited into the nucleus. It has a nuclear localization signal. If you have followed animal or plant biotechnology, you might be knowing what this NLS is. It's called as nuclear localization signal. Certain proteins, they have this signaling sequence at the end, which makes them, which tells the cell that they need to be localized in this particular compartment. Now in HTRT, it has a signal at the end which is localizing it into the nucleus. So the HTRT, which is made in the cytoplasm, it gets recruited in the nucleus. So it's important to consider at what time it gets recruited. It's during the S phase of the cell. We know during the S phase of the cell cycle only the DNA is getting replicated. So at this position, it's getting replicated and it binds with HTR, and there are some other proteins, reptin, pontin, which are important in the assembly of telomerase. So I hope you can see here now the HTR and the HTERT are both combined. If we take a look at the telomerase catalytic cycle, there are actually four steps. The first one is primer recognition and binding, synthesis of first telomeric repeat, translocation and realignment to the new 3' end of the DNA strand, and the dissociation. So let me explain you how the active site looks like. So as I said earlier, this is the ring-like structure which has a CTE, RT, TRBD domain. So this is actually the TERT or the catalytic protein part. There is another enzyme now. You can see there are these ones are there, right? TRBD, RT, CTE is present, but where is TEN? TEN domain is separate. Now, in this part, it corresponds to these three domains, but this TEN domain resides separately. So, what does this TEN domain do by residing separately? So, it has a very important function in the catalytic activity of the telomerase enzyme, which we will see in further. Now, here you can see the HTRT protein is there and also the HTR is there. So, HTR is the RNA template. So, the speciality of telomerase enzyme is that than other reverse transcriptase, it has its own RNA primer within it. So, sorry, RNA template within it. So, the primer for this would be the 3' overhang of the DNA strand, that is the natural primer for it. And if we consider the HTR part, the RNA part, it actually has three parts in it. One is this template, the other one is the pseudoknot, and the other one is the 5' boundary. So these three actually have reasons, these PK or the pseudoknot and the 5' overhang, these have reasons why they are there. We'll see them one by one. So this diagram depicts how the three domains of the TERT. I mentioned here that TERT part has four domains. So three are there as a ring structure. Earlier I said that it looks like the right hand form of a human hand. So the TEN domain is separate. What happens is now when the 3' overhang is there, let me draw it like this. If this is the 3' overhang, the telomerase enzyme comes and binds to it. So we know the sheltering protein helps it. So now we'll see very clearly and slowly how this happens. Right. So first, the 3' end of this binds to the catalytic site in the reverse transcriptase domain. Now what is this protein drawn here? It is the TERT. It's the TERT catalytically active protein. So in this, it binds to the domain which is reverse transcriptase here. So you can see the template is there, which is the inbuilt template which is here. So here comes and lies 3' end. So in order to hold this as an anchoring site, this TEN domain which is separate, this holds the 5' end of the template. So let's say that this is the DNA strand. So the 3' end is here and the continuous other DNA, the DNA would go like this, if it makes sense. So this is called as an anchor site. In research papers, they have mentioned a lot about anchor sites. So telling it simplified for you, the anchor site actually helps in the catalytic activity of the RT domain holding the template. So it helps in the positioning properly and also in the stability of the template in the 3' region and the region here, right? So what happens is in each step the nucleotide addition happens, we call it as a nucleotide addition processivity. The primary recognition and binding is done. So now the first telomeric repeat is going to be synthesized. How does it synthesize? The telomeric repeat, if you know, it is a nucleotide of 6 TTAGGG. So when these six nucleotides are being incorporated, we say that okay, one nucleotide, one telomeric repeat is done. So here, once those six are being done, one telomeric repeat is done, and this telomerase enzyme needs to add more, but it cannot add more since the 5' end, 5' boundary comes and meets. So in order, there should be a movement here. So that it could add more because the primer, sorry, the template doesn't move, we know the active site doesn't move, then how does this substrate moves with respect to the telomerase enzyme to add more nucleotides? We know that it doesn't add only 6 nucleotides at the end. What happens is there is where the 10 function comes. So 10 is an important protein, as I said earlier. TEN is to be said, as I read in research papers, it has a mobility activity, so it would move. When it is moving, this causes the 5' that is being positioned to move further, and the 3' to get positioned again, so that the telomerase enzyme could add nucleotides at the end to synthesize another repeat of telomere. So again, it synthesizes a TTA-GGG strand. So where this happens is in the 3'. If I go to this image once again, what it does is, in simple terms, it's extending this 3' end. What telomere does is it's extending this 3' end by adding TTA-GGT by many repeats. So it's elongating the telomere. Um, So this happens in 4 steps that I have said here. So the translocation step is where this TEN protein is being involved. Also, it's been said that another research paper says that there is kind of like a door latch, a way where once the enzyme is stick with the 3' end of the substrate, it continuously synthesizes the 6 nucleotides. Until then, the TN is in close proximity with the RT domain like a door latch. And once the 5' bound is met in the template, what it does is it would open the door latch configuration, and then it would release TN so that TN could move, pulling it this way, so that the 3' end can reposition in the proper place and synthesize another set of telomere repeat. So that's called the realignment. So like this, it could add many repeats. So what happens once the TN is moving? So when this is moving, we know that the templates then also could move because they have hydrogen bonds between them. So during this translocation, if we see, actually in the 3' this region, it causes a melting between the template and the substrate duplex. So the DNA bonds between them melt, and once they melt, uh, both the DNA strands unpair, which would be easy for TN to move. So once the TN is moving like this could also carry the template with it. So to avoid the template moving, it has this pseudoknot and the 5' boundary region. So these prevent the template from moving from the telomerase enzyme. So that's what the importance of pseudoknot and the 5' boundary has. They have more functions there, but basically that's what the idea is. So once they have synthesized such um, uh, telomeric repeats, what happens is at the end, the telomerase enzyme dissociate from the telomeric end. So this is the way primer binding, elongation, completion consists of a single telomeric repeat, and then this happened continuously, and at a stage it would dissociate. This is another diagram which explains it. So you can see that TEN has an anchor site here. This is the active site. The anchoring site helps on the mobility of the telomeric DNA primer with respect to the template because it needs to add more telomeric repeats. So now you might be having a question. What regulates how many telomeric repeats are being added into this end? Because if there is no control over that, the enzyme would continuously synthesize telomeres, or it could incorporate these nucleotides. So there is something called the telomerase. Give me a moment. We call it as the telomerase homeostatic system. So this homeostatic system regulates how long it needs to be. So at that point, the telomerase enzyme dissociates from the telomere. So this is the function of telomerase. So now we have learned about the enzyme structure. You know what the structure it is. Has a TR and a TRT, which means a catalytic protein part, and a RNA template. Now you know about the function. Uh, it elongates the telomere, add nucleotides to the telomere, so that once the telomeres are being added to it, even though the nucleus come and cut it, still the telomeres are only getting lost. The essential genes are not lost from the cells. And you know what's the mechanism which happens in 4 steps. I hope you understood something from this video. Thank you very much.